Lysosomal Disease Network did not get funded on the first go around. In fact, in 2003, our pink sheets said 3 different networks were hastily cobbled together to apply for the RDCRN1, but none were funded. And the bottom line on the pink sheets was you have a dream team, but we can't fund 3 different networks. So we spent the next 5 years, partially under NIH meeting support, to get together. And then over that 5 years, we congealed, if you will, a lysosomal disease network that was funded. The principle of metabolic cross-correction had been demonstrated to be clinically efficacious. There's some pioneering work, phase 1 studies by people like Roscoe Brain, Bob Desnick, John Behringer, and others, who are making great strides towards scale up. But really, at the beginning of that first cycle, there were only 2 existing therapies: Gaucher disease, enzyme replacement therapy, and bone marrow transplant. But then 5 years later, when we were funded, things had really heated up. There were about half a dozen therapies. Still, there are 99 lysosomal diseases we had identified, and none of them really had satisfactory therapies. So we set about to do the normal, natural history studies, and over the course of the 15 years where we have been funded, produced 171 peer-reviewed publications. As pointed out here to footnote 1, these are all NIH compliant, publicly available findings that accelerated the development of improved therapies rather. This was done by establishing a timeline of disease-specific behavioral and neurocognitive changes, protocols and outcomes for tolerizing regimens, new disease-specific biomarkers, quantitative brain morphology, methods for deep learning using artificial intelligence to analyze histology, biochemical quantification of lysosomal disease pathology, radiographic identification of CNS physiology, the outcome of some new drug treatments as they evolve, pharmacokinetic models for biologics like enzymes, methods of blood-brain barrier treatments, differentiation of male versus female clinical phenotypes. It goes on and on, also looking at comorbidities such as Parkinson's disease, POTS syndrome, and Clover-Bucy syndrome. So those are all laid out in the 171 publications. I will say that one of the things that our program officer told us from the very start, you're not going to have all the money you need to do what we really hope you will be able to do. So early on, after we had these ad hoc administrative meetings to organize a second application, and then initiated a research meeting called World Symposium, and then spun the World Symposium off as a separate entity, not at all funded by NIH. We got together. Those meetings are now about 2,000 people that meet annually in February. And we also want to thank Dr. Ed McCabe and Eva for publishing those abstracts in the meeting program in molecular genetics and metabolism every year. More recently, we spun off a patient advocate meeting called World Fair. Up until the present, it's been a locally run conference, and about 100 patients and others come along. I think this year it will go to a hybrid meeting, so people will be able to come to Minnesota where that patient-oriented meeting is located, but also spun out to the rest of the country. So I want to thank everybody, especially NIH, for all the great funding that's made this all possible. We've really drawn together the lysosomal disease community, and that means investigators, academia. It means biotech and pharma companies. It means the patient and some 65 patient advocate groups, as well as those even news broadcasters. So thanks very much for all the support, and we look forward to continuing on, perhaps as a nonprofit organization, Lysosomal Treatment Network.